Hello, my name is Keretz, and today I will be discussing my project on species distribution modeling of native unionid and invasive drusenid mussels in two large river systems of the Laurentian Great Lakes. North America is home to the highest diversity of native freshwater mussels, here in unionids, in the world. Focusing on the Great Lakes and the two large rivers in the Huron-Erie corridor, there were historically 39 species present in the St. Clair and Detroit River systems, including the now federally endangered northern riffleshell and snuffbox mussels. However, unionids were considered extirpated from the Detroit River shortly following the introduction of zebra and quagga mussels, collectively drusenids, into Lake St. Clair in the 1980s and the 1990s, respectively. Drusenids are particularly harmful to unionids because they can congregate on the unionid at high densities. This is known as drusenid fouling, which can interfere with the unionid's ability to feed, respire, move, and reproduce. In the Great Lakes region, drusenid fouling has been a major cause of unionid mortality. This, in addition to other harmful factors such as habitat degradation, are why unionids today are considered one of the most imperiled faunal groups in North America. Fortunately, there is hope. Unionid refuges, or places with living unionids and little drusenid impact, have been found in the Lake St. Clair Delta and in Lake Erie coastal wetlands such as Metzger Marsh and Crane Creek Marsh. Due to their highly imperiled nature, it has become increasingly important to identify and protect the unionid populations in these spatial refuges. One method for locating unionid refuge space is modeling known refuge locations. Bossenbroek et al. Were able to successfully predict unionid presence in Lake Ontario using Maxent unionid presence locations from Lake Erie and variables like fetch, shoreline geomorphology, and depth. Following up to this study, we are asking whether unionid presence can be similarly modeled in two large river systems, particularly the Detroit and St. Clair rivers. So, the first objectives of this study were to determine the species richness and relative abundance of remnant unionid populations, as well as the drusenid density throughout the Detroit River. We then used this presence data to predict potential presence of both unionids and drusenids in the St. Clair River. To start, we first determined a total of 60 sites throughout the Detroit River. 30 of those sites were randomly selected and are represented by the green squares on this map. 20 of the sites had been historically surveyed by either Schlosser et al. Or Kovalak et al. Between 1982 and 1998. These sites are represented by the orange squares. Finally, 10 of the sites were chosen based on our knowledge of unionid refuges and were classified as potential refuge sites. These sites are represented by the blue squares. We divided the river just above Fighting Island to create upriver and downriver sections, and all site types were selected equally between these sections. We set out to sample these sites between the 17th of July and the 9th of August in 2019 for a total of 18 days of sampling. At each site, we performed 3 10-minute dives with 2 divers for a total of 1 person-hour time search. We also took abiotic measurements such as depth, surface temperature, and secchi depth at each site. Finally, 6 petite ponar grabs were completed at each site to estimate drusenid densities and sediment composition. When any live unionids or unionid shells were found, they were brought to the surface, identified to species, and quantified. Live unionids were also measured to the nearest millimeter, and any attached drusenids were removed, stored in ethanol, and later quantified to species. Once we were back at the lab, we sieved the drusenid mussels from all 331 ponar grabs. Drusenids were then identified to species and quantified. Alright, so moving on to our results. We sampled a total of 56 sites throughout the Detroit River, and we collected over 2,000 unionid shells representing 31 species from 39 of those sites. These sites are shown here as white circles and white triangles. We did find live unionids at 5 sites in the lower half of the river at the sites represented here by the yellow circles. We found a total of 220 live unionids of 11 species. However, a majority of the unionids were found between sites 24 and 151, which were located immediately downstream from the Canard River. At just these two sites, there were 212 live unionids found, representing 10 species. It should be noted that only 4 of the 10 species were also found in the Clair River during its 2019 sampling season, indicating that the species found at these two sites were not just there as washout from the Clair River. Among the 5 sites with live unionids, the most abundant species found were the maple leaf, of which we found 91, and the fragile paper shell, of which we found 50. Moving on to dracenids, 51% of the unionids we found had dracenids attached. Sometimes that meant multiple dracenids, and sometimes it meant just one. However, even unionids that did not have a dracenid attached might still have shown evidence of dracenid attachment. In fact, only 2% of our live unionids did not have any evidence of dracenid attachment, meaning that the remaining 98% had either byssal threads or actual dracenids attached. The majority of dracenids fouling unionids were indeed zebra mussels, which are represented here by the darker blue bars. Quagga mussels, on the other hand, which are represented with the lighter blue bars, were only found on 3 unionid species. This is starkly contrasted by the dominance of quagga mussels throughout the river system. And this dominance of quagga mussels can be seen here, where we divided the dracenid counts by species and live individuals versus shell. Live individuals are represented in light blue and were determined to be alive at the time of the ponar grab by having characteristics such as intact periostracum, intact hinge, attached byssal threads, or intact tissue present. Alternatively, shells are represented in dark blue and were missing the necessary qualities to be considered, well, alive. We saw multiple outlying sites with extremely high densities of zebra and quagga mussels. For instance, at the sites with the highest densities, there were 826 live zebra mussels per meter squared and 5,674 live quagga mussels per meter squared. But if we stretch out these box plots, and I'll ask you to notice the change in scale on the y-axis, we see very low median values, just about 7 or 4 live individuals per meter squared. Such low medians came from the fact that most of our sites had very low densities present, sometimes even as low as 0. This is perhaps better shown visually. So, these maps show the relative densities of dracenids throughout the river. Live dracenids are shown on the left, and dracenid shells are shown on the right. In both maps, quagga mussels are represented in the darker blue, while zebra mussels are represented in the lighter blue. The small black dots that you see in the lower half of the river are sites where 0 dracenids were found. For both live and shell densities, we can see that there are higher densities of dracenids in the upper half of the Detroit River, especially when compared to the river's lower half. But perhaps the most important conclusion is that live unionids were found in the Detroit River more than 30 years post the dracenid invasion. So what's next? Well, now we are using the known presence locations for both dracenids and unionids to create species distribution models using Maxent. The environmental variables we considered were depth, total velocity, north-south velocity, east-west velocity, and river distance to the river's inlet, the river's outlet, and its nearest tributary. We ran multiple configurations of these chosen environmental variables systematically in order to obtain a best fit model for both dracenids and unionids. Within Maxent, I split the presence data into 80% training data and 20% test data. Each model creation went through 10 replicates using the subsampling and random seed parameters, ensuring that a different training set was used each replicate. The models were finally created using the default cloglog output. So, starting with both species of dracenids, the variables river distance to the outlet and the nearest tributary provided the best model with an AUC of 0.748. We first created a species distribution model for the Detroit River using our known locations of live dracenids. We then projected the model onto the St. Clair River. As a high probability of dracenid occurrence is represented by the red coloration, this model predicts a high chance of dracenid presence in the river's main channel, but a lower chance in the branches of the St. Clair Delta. For the unionids, the 3 environmental variables depth, total velocity, and river distance to outlet provided the best model with an AUC of 0.957. And like the Descendants, we first created a model for the Detroit River using our known unionid presence locations. We then used these known occurrence points to calculate a minimum probability threshold that could be used to predict areas that are likely to support unionid presence. So where our live unionids were found, the mean occurrence probability was 77%. The 5 sites ranged from 43 to 97%, with 4 of the 5 sites having a greater than 60% chance of unionid occurrence. We then projected the model onto the St. Clair River, and using that minimum probability threshold of 60%, the model considers 1.7 kilometers squared, or approximately 4% of the St. Clair River, to be good habitat for unionids. The model highlighted multiple areas like up near Sarnia and in the main channels of the St. Clair Delta. We are currently using these predictions to assist with selecting sites as represented here by the pink boxes. These sites will be sampled this summer along with a selection of randomly selected and historically surveyed sites. Similar to our surveys on the Detroit River, our systematic survey of the St. Clair River will be done with time searches and scuba diving. The validation of our models during this summer sampling will help us achieve our overall goal, which is to understand the distributions of Descendants and unionids in 2 large river systems of the Great Lakes. With that, I would like to thank our funding source and collaborating agencies. A quick shout out to all of the amazing people that have contributed towards this project. And if you have any questions, please feel free to reach out. Additionally, if you would like to keep up with our work, both in the St. Clair and throughout the Great Lakes watershed, you can follow myself and both of my advisors on Twitter. Finally, thank you for watching.